everyone, and welcome to this video which is looking at factors which affect enzyme action and also addressing some past paper question examples too. If you haven't already done so, please watch the video on enzyme structure first. You can access it by clicking the link at the top of your screen now. So I guess most of you will have covered the factors affecting enzymes in GCSE. You should know that there are four main factors you need to know about. The first one we're looking at is the concentration of enzyme molecules, and then we'll look at substrate concentration. After that, the two easiest ones to recall are pH and temperature. I want to talk about the concentration of enzyme molecules first. As you know, enzymes collide with the substrate molecules to form something known as enzyme-substrate complexes. In short, if we increase the concentration of the enzyme, we will increase the rate of the reaction. This is because lots of active sites are available. But if the rate of reaction is high, we will eventually find that we run out of substrate, or we can say that the substrate has become limited. If this happens, eventually there will not be enough substrate molecules for the enzymes to bind to, and the reaction will slow down. This example question here shows you how we can approach graph reading and describe and explain type questions for enzymes. So the question here reads, describe and explain the effect of enzyme concentration on the rate of enzyme activity. So what you've got here is two graphs, both of which show you enzyme concentration on the x-axis and the rate of reaction on the y-axis. The first one on the left shows you the increase in the rate of reaction as enzyme concentration increases. And so that's the first part of the describe question that you need to state. The second graph shows you how the rate would basically level off at some point when the enzyme concentration was still increasing. And this is where the substrate becomes the limiting factor. This is shown by the dotted line. At this stage, if you continue to increase the enzyme concentration, there is no further increase in the rate of reaction. The explanation part of the question requires you to say why. So why does it level off is the question we're trying to answer. You need to basically say that the substrate has been used up, or you could say that the substrate molecules have become product. You should further explain that if the substrate is in limited supply, less is available, and so the rate of reaction remains at a plateau. The next factor to look at is substrate concentration. We already know substrate molecules collide with enzyme molecules, and so there is a steady increase in the rate of reaction, as is shown in the graph on your screen now. The higher the substrate concentration, the more collisions occur, so the faster the rate of reaction. But, as you can see, the plateau is also happening in this graph. Where you can see that big yellow arrow pointing to, this is where the plateau starts, and this is known as the saturation point. This is basically where all of the active sites on the enzyme molecule have been used. They are known to be saturated, and so no more active sites are free for the substrate, so we see a steady rate of reaction as demonstrated by the plateau. So if we look at a describe and explain type question for substrate concentration, we've got a similar graph here. And what I've done here is I've split up the graph in three sections marked by A, B, and C in the purple pen. That first part where you see the upward part of the graph is where the substrate concentration is increasing, and you can see the rate of reaction also increasing. The point of saturation is at point B, and then you've got the plateau at C. This is where the graph is leveled off. In terms of explaining why that happens, so saying why that happens, well, we know at point A we have lots of free active sites on the enzymes and only very little substrate, so there are plenty of opportunities for an enzyme to collide with a substrate and the rate of reaction to increase. As we pass the point of saturation, we see that the plateau happens because the enzyme is limiting the reaction. There are so many substrates available, but not enough enzyme molecules, so only a few enzyme-substrate complexes will form, hence the rate of reaction levels off. The next factor we're looking at is pH. All enzymes have an optimum pH, or the pH that they work best at. Anything above or below their optimum pH is where the enzyme structure is disrupted. The hydrogen ions and the OH ions that are found in acids and alkalis affect the ionic and hydrogen bonds that hold the enzyme structure in place. Remember that the enzyme is a tertiary structure, so you do need to make reference to that. This means that the shape of the active site changes, and we say that the enzyme has become denatured. The graph here shows the typical bell curve that we would see when we look at pH against rate of reaction, with the peak of the bell curve being the optimum, as that is where the rate of reaction is at its highest. There is this table here that shows some typical optimum pH values for a few digestive enzymes just for reference. You don't need to memorize these pH values or know much about these particular enzymes, but you do need to understand that even in our body, there are such extreme pHs that occur, and so we do need different enzymes to carry out the roles in these areas. So, for example, our salivary amylase works best at an almost neutral pH in our mouth. We do have enzymes such as pancreatic lipase working at an optimum pH of around 9, and some proteases that work at a pH of 2 in the stomach. So if we do a describe and explain question, the first thing I would advise is for you to refer to the different bonding of the tertiary structure of the enzyme and how the changes in the pH will affect the hydrogen and the ionic bonding in that structure. You will need to explain that this causes the enzyme to denature and go on to say that the enzyme substrate complexes do not form, and so the rate of reaction falls, as is illustrated in the graph here. The final factor we need to look at is temperature. We already know from GCSE that an increase in temperature generally speeds up reactions. You must make reference to the kinetic energy of the particles. So as the molecules are exposed to higher temperatures, they move around more, and if they move around more, there are more collisions that take place between the enzymes and the substrates. As more collisions take place, there are more chances of enzyme substrate complexes formed, and therefore there is an increase in the rate of reaction. Nice and easy so far. The other bit that you do need to consider is that too much heat will cause a disruption in the bonds and therefore change the shape of the active site and cause denaturation. This basically means that the substrate will no longer fit the active site, and so the rate of reaction will drop, as it's shown in the graph on the screen now. So if we look at a describe and explain question, my advice would be to split your graph into 3 sections. The section where the rate is increasing, the peak or the optimum temperature, and then where it drops. In this diagram, I've labelled each part A, B, and C in purple pen. So the first part we want to describe is the bit between A and B. Here we see an increase in the rate of reaction, so we make reference to the increase in kinetic energy and increase in collisions, so lots of enzyme substrate complexes forming, and therefore the rate increases. All of these points will help you do the describe and the explain for this particular part. The next is looking at point B. To describe what's happening at this stage, we talk about the peak or the optimum being reached. To explain, we need to say that this is the best or the optimum temperature and the rate of reaction is at its highest point. And then lastly, we look at the describe and the explain between B and C. The rate here is decreasing, and that is enough for the describe part of the question. For the explanation, we have to say that the increase in the temperature causes the enzyme to denature and the bonds are broken in the tertiary structure. We can say that there is a change in the shape of the active site, and because the substrate no longer fits the active site, the enzyme substrate complexes will not form, and that's what causes the rate of reaction to drop. I hope that's nice and clear for you. So I think I'm going to finish up there for now. I do want to cover inhibitors next, so we're looking at non-competitive and competitive inhibitors, and I will also go through some past exam questions, as well as the required practical and the questions associated with that also. Thank you so much for watching this video on factors affecting enzymes. Bye for now.